well, good afternoon, everyone, and welcome to today's special fireside chat. I'm Amna Hussein, the Community Outreach and Engagement Manager at the Masonic Cancer Center at the University of Minnesota, and I will be your moderator today. The fireside chats are brought to you by the Office of Community Outreach and Engagement at the Masonic Cancer Center at the University of Minnesota. The Office of Community Outreach and Engagement serves as a vital bridge between the Cancer Center and communities across the state of Minnesota and beyond. Our dedicated team is committed to reducing the burden of cancer by actively engaging communities and providing access to relevant knowledge on the latest in cancer research, prevention strategies, the importance of screening and early detection, treatment options, survivorship, and opportunities for clinical research. Today's fireside chat focuses on the groundbreaking topic, how genome engineering is revolutionizing the fight against cancer. To guide us through this fascinating field, we are privileged to have Dr. Branden Moriarity as our speaker today. Dr. Moriarity is currently an associate professor in the Department of Pediatrics, the Division of Hematology Oncology. He graduated from St. Olaf College with a BA in Biology, Chemistry, and Biomolecular Sciences. He received his PhD in Molecular Cellular Developmental Biology and Genetics at the University of Minnesota. He then became a postdoctoral fellow in Dr. David Largaspada's lab, where he worked on identifying the genetics of pediatric sarcomas. He joined the Department of Pediatrics faculty in 2014. Dr. Moriarty leads a cutting-edge basic and translational research lab working on developing novel cellular therapeutics for gene therapy and cancer immunotherapy with the goal of translating new therapeutics to the clinic. To accomplish these goals, Dr. Moriarty's lab uses cutting-edge genome engineering technologies, including CRISPR-Cas9, which he'll cover today in his presentation. Dr. Moriarty is a recognized leader in cancer immunotherapy and genome engineering, and his contributions to advancing cancer research are truly remarkable. And so we are so fortunate to have him here today to share his insights and expertise in this emerging field. So before we dive in, I'll just cover a few housekeeping items. So today's fireside chat will be recorded as they always are and will be posted on the Masonic Cancer Center's YouTube page. For University of Minnesota employees and spouses participating in the university's employee wellbeing program, you can earn 200 wellbeing points by completing the form that will be linked in the chat shortly. And please use the Q&A feature at the bottom of your Zoom screen to submit any questions, comments, or your reactions that you have throughout Dr. Moriarty's presentation. We have time carved at the end for some Q&A. So without further ado, I will hand things over to Dr. Moriarty. The virtual floor is all yours.
All right. Well, yeah, thanks for the kind introduction, and excited to be here and share our work on advancing cancer immunotherapy using genome engineered immune cells. Let me just get a laser pointer here and we'll get going. So yeah, we are genome engineers. So maybe not all of you are familiar with this term, but you can think of it analogous to automotive engineers, where they're changing the parts and blueprints of a car to make it faster, more efficient, more effective. Now, we actually change the genetic code of living cells for the same purposes. And so you can think of the DNA of cells as the blueprint of how a cell is built and its components, and genome engineering gives us the ability to change those blueprints. And so we do this through either adding new genetic material or changing existing genetic material. And one of the exciting things about being here at the University of Minnesota is that both of these processes were initially partially invented here at the University of Minnesota. So Dr. Perry Hackett of the University of Minnesota was the first to develop what is called a DNA transposon. So this is a naturally occurring modality by which we can integrate new genetic code into cells, so we can give them new functions to do new processes. Additionally, Dr. Dan Voitas invented a technology called TALENT. This gives us the ability to change the genetic code of existing DNA within a cell to give it a new phenotype or function. This was shortly followed by the technology called CRISPR-Cas9, which many of you have probably heard of, which again allows us to change the genetic code of cells, which can lead to new phenotypes or functions in those cells, which can be applied to therapeutics. And so we really focus on cellular cancer immunotherapy. And so many of you may know that you have an immune system that's made up of many different immune cell types, and these have evolved to protect you from infections from bacteria, viruses, and the development of cancer. And in fact, when your immune system fails to detect cancer as it starts to develop, that's really when cancer is allowed to grow and thrive and becomes clinically detectable. Thus, it seems intuitive to me and many others in the field that using these immune cells that have actually evolved to detect and eliminate cancer as a therapy is a really intuitive idea. Now, saying that, it is unclear which immune cell type would be best for cancer immunotherapy because many of these different cells are involved with the process of identifying cancer and eradicating it as it evolves in a patient. And so some people have used a subset of T cells called alpha beta T cells. These are so called very personalized therapies or autologous therapies because you can only use a patient's own alpha beta T cells for their therapy. If you were to take someone's alpha beta T cells and put them in another patient, they actually would see that patient's tissue as foreign and attack those cells. Now, there are other immune cells that we term off the shelf therapies or allogeneic therapies. So, these are immune cells where you can take them from a healthy donor and put them into a recipient, and they won't attack the recipient's tissues. And so, these are two different cell kind of populations that we can use for cancer immunotherapy, but it's still very unclear which of these different subsets of immune cells will be the most effective for cancer immunotherapy. And so, more recently, cellular cancer immunotherapy has really focused on isolating some sets of these immune cells, engineering them in the laboratory, expanding them to very large numbers, and then reinfusing them into the patient. And so, we've seen actually miraculous cures in hundreds of thousands of patients, particularly patients with leukemias, using this type of approach. And so on this slide, what I'm showing you is one of the most prevalent cancer immunotherapies that's available today. It's called CD19 CAR T cell therapy. And so what they do here is they take out the patient's alpha beta T cells and they put in new DNA, new genetic code, and that allows the cell to build what you can think of as a metal detector. And this metal detector allows that cell now to identify the cancer cells and eradicate them directly. And without this metal detector, these T cells really struggle to identify the cancer and eradicate it. And so here on the right is a survival curve of some of the clinical data using this approach for leukemia, where normally only 10% survive past 5 years. And you can see here almost 80% of patients survive more than 4 years now with this CD19 CAR T therapy. And this was actually the first FDA approved cancer immunotherapy called Chimera, and now many others have followed, looking at CD19 and other tumor-associated markers. Now, an older technology, but very, very promising, is called tumor infiltrating lymphocyte therapy or TIL therapy. And so this was developed by a researcher at the NIH called Steve Rosenberg. And he had this theory that the immune cells that are found in a tumor are probably there for a reason. They probably are there because they're trying to fight the cancer, but they're losing the battle. And so he came up with this process where he would actually remove the tumor and then he in the lab, he would cut them into small pieces and then grow out those T cells or those immune cells. And then he had an ability to screen them to see which ones were actually effective at fighting the cancer. He would grow those up to millions and billions in the laboratory and then reinfuse them into the patient. And so this is again called TIL therapy. And this is the only therapy in history that has cured highly metastatic solid tumor cancer. The only problem is that it doesn't work for all patients. It works really well in melanoma and non-small cell lung cancer, but it hasn't worked as well in other solid tumor cancers like GI cancer or breast cancer. And so now we and others in the field are using genome engineering to also further enhance this type of therapy called TIL therapy. And so, with that said, now I'd like to give you an overarching view of our laboratory, what we work at called translational genome engineering. And so, this is really advancing cancer immunotherapy using genome engineered immune cells. And we really see our translational pipeline, kind of from concept to clinic in three different phases. So, in the first phase, we really identify the cellular type that we would like to engineer for a given cancer indication. And we pick that cell type given different attributes of those cells and the type of cancer and what we think might work best. And so, in the first stage, is where we actually optimize our ability to change the genetic code of those cells. So, our ability to introduce new DNA to give them new functions or alter the DNA of that cell to give them new functions. Moreover, what we've learned over history of cancer immunotherapy is that you really need to deliver many, many of the cells for them to be effective. And so we also optimize our ability to grow these engineered cells to very large numbers. And luckily, from work from our lab and others, many of the stage one events for these different immune subsets have already been worked out. So we really have the ability to engineer most of these immune cells with very high efficiency and grow them to very large numbers for therapeutic use. Stage two is where we really start to partner with clinicians on specific cancer indications or with companies and really decide what kind of genetic edits would make this cell the best for a given indication. For instance, for breast cancer or lung cancer, how could we deliver specific CARs or those metal detectors to detect the cancer? And then what genes might we turn off or on in the cells to make them more effective against that cancer? And then we basically build a prototype of that genome engineered cell, and we do preclinical testing where we ask the questions like, is this really effective against lung cancer or breast cancer? And then we'd use mouse models where we actually implant human tumors in the mice, and we deliver our genome engineered cells to the mice, and we ask questions like, are they effective? Can they find the tumor? Can they eradicate the tumor? And also, is it safe? So does it harm the mice? Is it toxic to the mice? Things of that nature. And so that's stage two. And when we find something that's really exciting and we think it could have good efficacy in the clinic, that's when we move to stage three of the process. And so this is where we work with what we call good manufacturing program facilities. And we have one of these at the university called the Molecular Cellular Therapeutics Facility. And so you can imagine the cells that we're making in the lab, we're not allowed to actually put those in people because we don't have all of the manufacturing background and paperwork and tracking and things of that nature. And so at these special GMP facilities, that's where they can actually manufacture in sterility these products that can go into people. And so basically, we train them how to do the genome engineering that we've worked up in the laboratory, and they perform these process validation and qualification runs that basically shows that they can generate the exact same cells that we developed in the laboratory, but in a setting where they can be used in people subsequently. And so then we take all of that data from stage two and three, and we file for what's called an investigational new drug application with the FDA. And so they will either tell you, okay, you've provided enough safety and efficacy data that you can move in to a clinical trial to treat patients, or they might have additional questions and data requests that we then generate until we get FDA approval to initiate the clinical trial. And so, using this three-stage method, we've actually launched six clinical trials over the last 10 years for both blood cancers and solid tumor cancers. And today I'll give you a couple examples of those trials and kind of how they came to fruition and some of the results of those trials. But before I jump into those vignettes, I just wanted to touch on a little bit about the funding and strategic partnerships that allow us to get these clinical trials launched. And so you can imagine that this translational work is quite a costly endeavor, requiring several funding sources. And so we do, avail of ourselves of many of these, including funds from the National Institute of Health, the Department of Defense, foundations and philanthropic organizations. We also have partnered with industry for many of these. So on the right are just a list of some of the different companies that we've worked with to actually launch these new therapies into clinical trials. And so what we and others have found is that depending on the complexity of the clinical trial, it can cost anywhere from $2 to $10 million per trial. And that's why we really needed to establish many of these industry partnerships to really take these things from concept to clinic. And as we are developing these new therapies, we're also developing intellectual property that's owned by the University of the Minnesota. And many of those have been licensed to companies that have then taken that technology into clinical trials for cell and gene therapy as well. And so that's one avenue where we can have an impact on getting these into the clinic, but not directly do all of the different stages ourselves, but kind of hand that off to a company that can do it effectively as well. The other thing that we've done to leverage our ability to get more of these cell therapies into the clinic is started new companies, what are called U of M spinouts. So they're companies that are derived from the University of Minnesota that license intellectual property from the University of Minnesota, and then they go on to start clinical trials and really translate these therapies into the clinic. And so myself and others in the Center for Genome Engineering have been involved in starting many of these companies. Again, as you can see, using many different immune cell types for different indications, because again, we really don't know which cell types are going to be most effective for cancer therapy. And so here's just an overview of the ongoing kind of concept of clinic trials or products that we're developing in the laboratory. And you can see some of them are led by the U of M and carried out here. Some of them are out licensed, like in green there to Beam Therapeutics or Catamaran Bio. And then some of them are from U of M spinouts like Luminary Therapeutics, who I'll tell you more about their clinical trials today. But there is, many challenges still in this adoptive cell therapy space or the cellular-based immunotherapy space. So still, many of these therapies are highly personalized. So as I mentioned, many of them use those alpha beta T cells, they're autologous therapies, which makes them a very complex manufacturing process and can be very expensive. Moreover, when they actually bring in new genetic code or new DNA, a lot of their approaches are using viruses, which are very randomly integrating, can have safety concerns and cost and complexity as well. So that's another challenge for this field. And then, moreover, the solid tumor space has been a consistent challenge for adoptive cell therapy. There have been some wins, but many failures, much more successful in liquid tumors like leukemia or lymphoma. So some of the challenges in treating solid tumors are the homing of the immune cells and infiltrating the solid tumor. They also see this phenomenon where solid tumors will kind of lose their cancer markers, so they kind of trick the immune cells. And then these solid tumors develop very immunosuppressive microenvironments. So these tumor cells purposefully will make things that suppress the immune cell's function. And so solid tumors have been a very challenging cancer type for both cell-based immunotherapy and other therapies. And so some of our approaches to enhance these adoptive cell therapies or immunotherapies is really testing many different immune effector cells, because again, we don't know which ones are going to be really the kind of linchpin or winners. And then in our case, we apply genome editing broadly for precise editing and multiplex editing, as you'll see in some of the data I'll show you. And then instead of using randomly integrating kind of viruses to bring in DNA, can we site specifically bring in new pieces of DNA in a more safe fashion? And then we also apply this genome editing for functional enhancements. And so making these therapies more of the off-the-shelf type that I was mentioning, so you can make large batches and apply them to many different patients, and then making them armored or resistant to the solid tumor microenvironment, those suppressive signals I was talking about, and then also programming them through adding or modifying DNA so that they can better home to and migrate within solid tumor cancers. And so one of the kind of mainstays for classical genome engineering that is used today is called CRISPR-Cas9. So I think many of you have probably heard of this technology, but essentially it's a programmable enzyme. And so it's this enzyme that we can program to bind any sequence of DNA in living cells. And essentially, it does a very simple thing. It binds to the DNA and it breaks the DNA in half. And whenever the DNA breaks within a living cell, we've evolved mechanisms to repair that DNA. You can imagine if the blueprints of your cells get broken, it's a bad thing. And so we have these mechanisms to actually fix them. Most of the time, when the DNA is fixed, they basically just take the two broken pieces and put them back together. And that a lot of the time does correctly repair the broken DNA, but sometimes it doesn't. And when it doesn't, it can lead to uh, inactivation or knockout of that gene's function. So this way we can use CRISPR to actually knock out genes' functions. And so if it's a gene that suppresses the immune cell's function, then we can turn that off by breaking the DNA. Another thing we can do is actually we can provide a piece of DNA that we can trick the cell into inserting in its genome, a process called homologous recombination. And so through this, we can actually put in new genetic code to give the cell new functions. And so we actually utilize CRISPR-Cas9 in a TIL therapy, that therapy I mentioned earlier. And so I'm just going to walk you through some of the work that we did for the stage 2 and 3, kind of the bench to bedside process. And so this is all published, so I'll go over it at a high level and you can read the article if you like, but for now you can take my word for it. But essentially what we found working with Dr. Steven Rosenberg is there was one specific gene that was activated in these TIL that actually suppressed their ability to fight cancer. And so what we did first is we confirmed this observation in human TIL. And so we took these TIL from human patients and we found that indeed this gene called CISH was upregulated in these TIL, these T cells that were in the tumors. And so we also showed that they indeed were suppressing their function for cancer immunotherapy. And so then what we did is we developed a CRISPR approach to actually turn off this gene or break that DNA of CISH. And so this data is just showing that our ability to get rid of the gene is very good. So this is called a Western blot. And what it's showing is the protein that is derived from this gene is still there in our control sample. But when we use CRISPR, it's completely gone from almost all of the TIL in the population. We also showed that this genome editing approach was very specific and very safe. It did not break the DNA in the genome anywhere else except at the cis gene. So that's very important. We further went on to show that if we took these T cells and we got rid of the gene CISH, that T cells were better at killing cancer and higher functioning, and they were better at identifying and being activated to kill the cancer, just in T cells. And then we actually took TIL from patient tumors and we got rid of this gene CISH and again showed that they were basically better, faster, stronger at attacking that cancer and eradicating it. And then lastly, we performed that stage 3 component where we took our ability to do this genome engineering, we brought it over to our GMP facility. They generated their process validation qualification runs showing they could actually do this in their facility. And then we took all of this and filed for an IND, which was approved by the FDA. And so this was the first in human phase 1/2 clinical trial using these CRISPR engineered TIL, and we were actually treating metastatic GI cancer. And so this is a kind of a basket trial for GI that includes pancreatic, colorectal, cholangial, many different types of cancer. And so here we worked with the clinician Emil Liu and my co-PI investigator Bo Weber. And so essentially patients would come in, we would remove their tumor, we would isolate their T cells, and then we would CRISPR engineer them and infuse them back into the patients. And we actually just finished this trial about a year ago, and we're publishing the results from this. But just at a high level, all of these patients are coming in with rapidly progressing, highly metastatic cancer. This is basically their last therapy that they'll get to try. And what we found is that about half of the patients we treated of 12 patients, they had stable disease at 1 month. So we actually halted the growth of their cancer. And a third of the patients, their cancer stopped growing for 2 months. And then all of those 11 patients progressed, and all of them have succumbed to their disease, unfortunately. But we did have one patient, which we call UMN 22, that had metastatic colorectal cancer. And after about 4 months, all of her cancer melted away and disappeared. And she now has a clinical complete response with no cancer, more than actually about 14 months now, devoid of cancer pain, back to living life with her three young children. So this was a pretty exciting result to actually cure one person with highly metastatic colorectal cancer. Again, showing the promise of this TIL therapy can work in some patients, but we likely need additional genome editing to make this more effective in more patients. And so that's some of the work that we've done clinically using Cas9 enzyme or nuclease. And so the other story that I'll tell you here is using a slightly different genome editing technology called base editors. And so this technology kind of evolved because sometimes people want to edit more than one gene or insert DNA in the genome in more than one place. And unfortunately, when you do this with this Cas9 that breaks DNA, on the right over here, you can see this little cartoon where if you break the DNA in more than one place, the DNA chromosomes can actually what we call translocate, or they can move to other chromosomes, which can be very toxic to cells and can actually lead to transformation of that cell, or that cell itself can turn into cancer, which is not good. So basically, if you're using this Cas9 technology, you can do one genome edit very safely. But if you want to do multiple genome edits, it's not a great technology. And so, how can we perform multiplex or multiple spots in the genome editing without breaking the DNA or inducing these double-stranded breaks? And so, luckily, this investigator at the Broad Institute named David Liu, he made a subtle modification to this Cas9. And so, what he did is he removed the enzyme's ability to break DNA, but it still retained its ability to be programmable to bind anywhere in the genome. And so what he did, which was super clever, is he fused that enzyme to another enzyme that actually changes DNA letters. So your genome is made of C, T's, A's, and G's at the genetic level. And so with this first tool, it gives us the ability to change C's to T's in the genome. So basically making subtle modifications to our blueprint that still can lead to changes in functions of the cell. He later developed another technology that allows us to change A's to G's in the genome. You can kind of think about this like computer programming, where they use ones and zeros to program a computer. We use A's, C's, T's, and G's to program a cell. And so this allows us to change the genome in many places without having those dangerous double strand breaks or breaking the DNA. And so this tool allows us to make many changes at once very safely in the genome. And so we first deployed this in making a CAR T cell product, kind of like that CD19 that I was telling you about earlier. And so we wanted to actually edit three different genes at one time. One was a gene called PD1. And what this does is it allows cancer cells to suppress these immune cells from doing their job. And so we wanted to go in and turn this gene off. And I remember at the beginning, I was telling you these types of T cells, these alpha beta T cells, they can't be used from one patient to another. But researchers have found if you turn off two genes, one called a T cell receptor and one called B2M or HLA, you now can use these cells safely in one patient to another. And so by turning these two genes off, you remove that ability of the cell to attack the patient's tissues, called graft versus host reaction. And then it also removes the patient's own immune system from being able to reject these therapeutic cells. And so we did these three gene edits. And then we also introduced one of those CAR molecules. And in fact, it was that same CD19 CAR that I told you about earlier. And so again, this is published, so I'm just going to go over this at a really high level. But essentially what we found is that this new tool, this base editor, allowed us to edit 3 genes at one time with very high efficiency. Actually, 90% of the T cells had all 3 genes edited simultaneously, which was pretty incredible. Moreover, we looked at the safety of this. And so we wanted to see, does our new tool accidentally break DNA? And in fact, we found it does not. But we did again found when we used one of those Cas9s, or the ones that break it, we actually found that the chromosomes were fusing with every single break that we made, which is definitely not what we're looking for. That can transform cells. It's very dangerous. But this new tool, this base editor, allowed us to do that safely. We also found that when we used those breaks, the multiple breaks, it actually affected the cells so much it slowed their growth, and we weren't able to grow enough to make therapeutic doses. But we found that when we use this base editor technology and we turned off those 3 genes, they were even more effective for cancer immunotherapy. And so, based on this work, our intellectual property from the U of M was licensed to a company called Beam Therapeutics, and they have now taken this into the clinic for actually T cell leukemia and lymphoma, and multiple patients have been treated under this trial that is still ongoing. And so the last part I'm going to tell you about actually doesn't use the CRISPR-Cas9 technology. It uses that first technology that I presented that was first developed by Perry Hackett, that DNA transposon technology. And so the way that these work, they're a naturally occurring genetic system, and they are composed of 2 different components. One is basically a circular piece of DNA. So these are kind of like new blueprints that we can add to a cell to give them new function. And then there's another component called a transposase enzyme. And essentially, this enzyme will identify the genetic cargo or the DNA, and it will randomly integrate it into the genome to introduce new DNA code to give that cell new function. And so we build these 2 components, we deliver them to cells where our DNA cargo is integrated into the genome by our enzyme, and then that will produce new proteins and new capabilities for that cell to make it better for cancer immunotherapy. And so actually, that first technology that Dr. Perry Hackett developed, it was called Sleeping Beauty transposon, and that had been licensed away to another company, and basically no one had access to that technology anymore. And so what we did at actually one of our first U of M spinout companies called Beamogen Biotechnologies, we wanted to build a new transposon system that could be used broadly. And so what we did is we looked to nature because these things are naturally occurring, and we found a new transposon called TC Buster. And this had been previously identified by a researcher named Nancy Craig, and this was her paper on this. And she showed that this indeed could work in human cells, but it didn't work very well. It was very inefficient at inserting new DNA into the genome. And so what we did at Beamogen is we actually looked at other transposons in nature, and we started putting together different components or different amino acid sequences, and then we would screen them to see how efficient they were at inserting DNA into cells. So we made lots of different variants, and we found that when we changed some of the code of this enzyme, it did get better at inserting DNA. So like about 30% of the cells would get the DNA inserted in the genome, but we wanted something that was much more efficient, like 70, 80% of the cells receiving this new genetic code. And so we did kind of a sophisticated screen where we actually randomly mixed up this enzyme's code. And then we made 5 million variants of this enzyme. And then we screened them through kind of this sophisticated screening mechanism. And then we found all these different enzyme variants. And in the end, we found one that was very efficient. And in fact, it could deliver DNA material into almost 80% of cells that we treated with this DNA transposon and transposase enzyme. And so this was actually acquired by another company and now is provided globally through licensing to anyone that wants to use this technology. So this is truly available to anyone that wants to use this tool to insert DNA into cells to change their function for therapy. And so after we had that spin out company, we started another one called Luminary Therapeutics. And here we actually utilized that TC Buster transposon technology that was now available, and we applied that to engineering immune cells for therapies. And so what we did is we developed a different CAR. So the first one, that metal detector I told you about, identified something called CD19. And this metal detector identified three different markers on cancer cells, which is really ideal because these cancer cells are very tricky. And if they get rid of one of their markers, then they become invisible to these CAR T cells. And the kind of crafty thing about this one that we built, this BAF ligand CAR, is it identifies three different markers on cancer cells. And so for a cancer cell to escape from our therapy, they would have to get rid of three different markers on their cell surface, which is really, really hard for them to do. And so we think this is going to be a better therapy than things like CD19 therapy. So then we essentially carried out the stage 2 and 3 process to get this into the clinic. And so we showed that we could engineer T cells with very high efficiency. We showed that when we inserted the new DNA, it was very safe. It wasn't like disrupting the genome or messing up the blueprints in some way. We then tested it in vitro and in animal models, and we showed that it was really effective against mantle cell lymphoma, multiple myeloma, and acute lymphoblastic leukemia. So against multiple different kinds of leukemia and lymphoma. We also showed that it was safe, so it wouldn't kill normal cells and it didn't hurt mice. And then finally, we published this data in a primary manuscript and we got IND approval to start our clinical trial in less than 18 months. And so to date, we've actually treated 4 patients. Here's the results that I can share with the first 2 patients. So the first one had transformed follicular lymphoma, and that patient at the lowest dose of our cell therapy actually had a complete response, completely cured of cancer. Our second patient had stable mixed response, so a lot of their cancer went away, but not all of it. And again, this was at the lowest dose of the T cells. And again, both of them, they were very safe. Neither of them had adverse events from this therapy, which can happen with some types of immunotherapy. So we're pretty excited about this therapy. Just one side note, not only is this therapy effective for cancer, but now we're actually also applying it to other indications such as autoimmune disease. So now we and others have found that you can treat things like systemic lupus with these engineered immune effector cells. And so now we actually have an active IND to treat people with systemic lupus with this BAF CAR T cell product to treat their autoimmune disease. And so that's kind of the different vignettes that I wanted to share about some of our clinical trials and what's still going on. But I wanted to leave you with one huge unmet need in our translational pipeline. And that is essentially that stage 3 of the translational process. So I can say lots of the discovery and therapy development is really well funded through NIH, DoD, and foundations. Everyone gets really excited about new discoveries, making new therapies, testing them, but they see stage 3 as kind of boring. It's kind of like this necessary evil. It's work you have to do to actually get it into the clinic. And so people that are used to funding, innovative discovery science, they see it as boring and they don't really want to fund it. And so again, here's that diagram of that stage 3 where we kind of show the GMP facility how to do the engineering, they scale it up. There is essentially zero funding for this stage of the endeavor. And so this is a huge unmet medical need or funding need, and I just wanted to share that with everyone. Um, the later parts, the actual clinical treatment of patients, there is also reduced funding there, but it's getting better. We're starting to see some new funding mechanisms from both NIH and DoD that will actually pay for the treatment of the patients with novel therapies. But again, this process of taking it from our lab into the GMP manufacturing is a huge unmet need, and it really is very expensive. So just the first phase is almost a half a million to $1 million for the data they generate to get the IND. And then again, that clinical trial can cost anywhere from 2 million to 10 million, depending on the complexity of the therapy. And so with that, just some conclusions and future directions. So here at the U of M, we developed and translated CRISPR-edited TILs for GI cancer, and we carried out that clinical trial. I forgot to mention that that was actually the second FDA-approved CRISPR trial in the country, which was pretty exciting. We've developed these methods for multiplex editing of T cells, so we can modify many genes in the genome at once, and that was translated and out-licensed by this company, Beam Therapeutics. We developed and translated a novel DNA transposon technology. And then again, finally, the stage 3 of translational genome engineering is a huge unmet funding need. Future directions. So we're really continuing to push the envelope of non-viral and multiplex genome engineering to really give these immune effector cells all the tools they need to truly cure solid tumor cancer. And some of the mechanisms we're using for this is that we're doing things like armoring them to kind of protect them from suppressive and shifty cancer cells. We're engineering them so they're better at homing to the tumors and identifying those and infiltrating those. And I talked about these metal detectors and how they help them identify cancer. We're actually engineering immune cells so they have multiple metal detectors, so it's harder for the cancer cells to sneakily kind of evade our engineered cells. And then, of course, we continue to identify new academic and industry partners to translate additional cell therapies for cancer and now even other diseases as well. And so with that, I just want to thank all of our funding sources and all the people in the lab and our collaborators. Particularly large shout out to Dr. Bo Weber, who helps with all of this work. We're co-principal investigators on all of this work, and we run a joint lab here at the University of Minnesota. And with that, I will stop and take any questions.
Thank you so much, Dr. Moriarty, for your fascinating presentation. I mean, it's so inspiring to hear how this cutting edge genome engineering technologies at the University of Minnesota is paving way for innovative cancer research and treatment globally. So thank you for all that you do. And so we will now transition to our Q&A. So this is your opportunity to engage directly with Dr. Moriarty. So please submit questions that you have, any comments that you have. This is your chance to do that. And as all of you know, if you participated in a fireside chat before, it's not a fireside chat Q&A without getting our fire started. So let me just set the mood with our virtual fire. All right. Okay, so as folks are submitting their questions, I'll start off with some of the questions that have been submitted already. So how are advancements in microbiology and especially our understanding of the microbiome affecting work in immunotherapy?
Yeah, that's a great question. This is quite an emerging field, actually, where people are finding direct correlations with the microbiome and outcomes of immunotherapy and other cancer therapies, where the makeup of your microbiome can actually affect the efficacy of things like checkpoint blockade and even CD19 CAR and other CAR therapies. The mechanisms behind those are still being ferreted out for the large part, but the connection is true and real. And again, it's just like a blossoming field where people are trying to understand it. It could be that one day you even are taking, some sort of probiotics that are engineered themselves to facilitate or enhance a cancer immunotherapy to work kind of in synergy or concert together. So it's a very fascinating kind of emerging field, definitely.
That's definitely really interesting to see how these fields are potentially coming together and how they can enhance some of our treatment options. Okay, so I see a question that's just been submitted. Are biopsies and other samples collected from patients in clinical trials you discussed for subsequent research to investigate why some people fared better than others?
Yeah, definitely. A great example is our engineered TIL therapy, where the GI cancers where we took out their tail, engineered them, put them back in. So there, after and before the therapy, we took blood samples, basically every couple weeks. And that's allowing us to start to ask why some of those patients weren't cured of their cancer. Was it that the immune cells that we put into those patients, they didn't persist a long time? Or was it once we put them in there, they became very tired and dysfunctional and were unable to fight the cancer? Which gives us clues into how we could improve the therapy in the future. Because if we find that they're not persisting, for instance, we can engineer in new capabilities in those cells to allow them to persist longer, to kind of give them a, a longer battery life, if you will, to persist in the patient and give them a better chance of, finding and fighting the cancer. So certainly all of our clinical trials were collecting associated biopsies, blood samples to really look at mechanisms of resistance or mechanisms of why that therapy was not effective in that patient, but maybe it was in another patient. So that's definitely something we're moving into more as our clinical trials are ongoing and wrapping up. We'll be working on more of what they call correlative studies or those investigational follow-on studies.
Awesome. All right. So will these techniques be used to treat cancers driven by mutations like BRCA to repair the mutations?
Yeah, that's another good question. So I described this kind of CAR T technology where they give them that metal detector and they can find them. That is quite a broad metal detector, but we can engineer them with a different kind that are more specific to a given individual, and those are called T cell receptor therapies. So basically, I'm trying to think of a good analogy here, but it's kind of a metal detector that doesn't work with every kind of metal. That's a good way to put it. So these cars, they can find copper and they can find lead, but these other ones, they can only find lead, let's say. And just as an analogy, and so every person has a different type of metal or a different background, if you will. And so these TCR kind of therapies, they can't be used in all patients, but some of them. And there they can be looking at specific mutations. And so the way that that works is every cell kind of displays a little bit of what's going on inside their cell on their cell surface. And so with these TCRs, they can actually find these mutations, these little mutant bits that are on the outside of the cell. And so those are specific down to the exact mutation. And we do have some projects working on those types of therapies. They're just a little more personalized in that way, but they do have high potential for various reasons that are kind of technical. And so, yes, the answer is there are therapies that are specific down to the mutation of that given patient.
That's great to hear. So, what are some of the most significant challenges that you faced in translating these therapies from the lab to clinical trials, and how are you addressing them?
Yeah. Again, it's really that stage three of the translational process where it's kind of a necessary evil or something kind of boring and not discovery science that institutes like NIH and DoD really aren't interested in funding right now. And so that's where we look to philanthropy and also industry partnerships. And some foundations will even fund some of that work, but that's definitely the whole, or some have termed it the valley of death of getting
Mhm.
something that's really exciting from the lab into the clinic is doing that kind of phase or stage three work and getting that data you have to get the FDA to give you approval to start your clinical trial. So that's definitely the largest hole in the process.
So are HeLa cells part of this research, or have they impacted your work, and how so?
Yeah, so none of what I showed you today specifically uses HeLa cells, but HeLa cells is a model cell line, of course, derived from Henrietta Lacks, that had been kind of a really awesome tool early on in the field of studying cell biology, cancer biology, because we didn't have a lot of cancer cell lines that we could grow in addition in the lab. Luckily, over the years, people have identified different growth conditions, medias, and approaches to grow many different types of cells in the laboratory. So now we have the choice of thousands of different cell lines from hundreds of different cancers that we can use in our preclinical testing. Some people still use HeLa cells. They definitely are still a useful cell line to work on. We just haven't used them in any of this work specifically.
All right, so keep the questions coming. These are great. So I know, so you mentioned the importance of funding to do this innovative research. What tips do you have then for new investigators seeking funding?
Yeah, that's a great question. I mean, when I started out my career, you start small, right? Be very focused and go for the low-hanging fruit grants. Like for instance, NIH, they have really large awards, for really established investigators, and they have kind of smaller introductory grants that you can go after. Also, philanthropic investment in your laboratory is really important, particularly in the beginning, so you can use a small amount of money to generate some exciting data that allows you to really go to NIH or DoD to get those really high level grants. So, really interacting with philanthropic institutions that are in your field of study or cancer of interest, I think that's a really good way to kind of get your career going is, looking for those small investments, building data sets that can get you those large investments.
Thank you for that. So, can you give us kind of a timeline of when some of these engineered therapies will become widely available and accessible?
Yeah, I mean, we're living in a very exciting time. This field is now, 20, 30 years old. And so some of these therapies are becoming actually standard or first line therapies for some of these cancers. CD19 CAR T cell therapy, there's been a number of them that are fully FDA approved. Like the patient comes in with leukemia and the doctor can literally order CD19 CAR T cell therapy. More recently, the FDA also approved the first two TIL therapies. So that's for melanoma and non-small cell lung cancer, where that therapy works for a lot of the patients. Like TIL therapy works almost in 50% of patients treated with that therapy, which is incredible. So we're really seeing some of these coming to fruition where they are becoming more and more early kind of first line therapies. But it's going to be probably, another 10, 20 years before, many different cancer types have a first line or second line therapy that's a cell-based therapy. So it's an evolving thing, but luckily we're in an exciting time where some of them really are already coming to fruition, but there's still a lot of work to be done, particularly in solid tumor cancers that are much more challenging.
Thanks for that perspective. All right, so clinical trials. So are there current clinical trials that are available for folks to enroll in, and how can they find out if they enroll?
Yeah, definitely. I mean, there's literally
They qualify.
hundreds of new clinical trials across the country and across the world. Obviously, if you are suffering from cancer, working with your primary care oncologist, but you can also go to a website called clinicaltrials.gov, and you can search different cancer indications and see what kind of therapies exist. And there are literally hundreds of these cell therapies, small molecules, antibody therapies for various indications. But definitely, work with your doctor. You can go and find, new options and opportunities yourself through this clinical trials website, but then definitely, coordinate with your physician and see if it's the right thing for you at that time. But yes, there are lots of opportunities for lots of cancer indications.
Great. So I just put in the chat clinicaltrials.gov. Thank you for that. So how can the public support this research and address some of the challenges that you mentioned?
Yeah, I mean, like I mentioned, for even new investigators and established investigators looking to investigate or develop new therapies, getting small seed money from either granting organizations like Some of the local ones here are like Children's Cancer Research Foundation, things of that nature. But that seed money that you can provide for this type of research is super critical. That allows us to go and get huge grant funding from NIH that is required to kind of get us through stage 1 and 2, really develop something and vet it out that, we know it's exciting. We want to test this in people. And so I would say either donations to local philanthropic organizations or direct donations to investigators' labs, which you can do through the University of Minnesota foundations here. Those are direct approaches to actually support this type of research.
Great to know. Okay, final question for you. So Dr. Moriarty, tell us what excites you most about this field?
Yeah, I truly believe that this is going to be the cure for all cancers. That's what excites me the most. Our instance where we were able to cure some with highly metastatic colorectal cancer was like, one of the highlights of my first 10 years of my career, and we hope to do that for many other patients. And so I think just this promise or this potential that we have a lead onto something that could be the cure for all cancers, and it really just needs to be optimized and perfected for given indications. I think that's really exciting. The other thing that's exciting is now we're able to take these same technologies and apply them to other diseases and indications like autoimmune disease. And so I think broadening the scope of these engineered immune effector cells into other diseases and disorders is also super exciting.
Incredible. So thank you for all of your work. But we've reached the end of our time, unfortunately. So thank you all for joining us and for engaging with us today. And again, special thank you to you, Dr. Moriarty, for sharing your expertise and for just giving us a glimpse into the future of cancer treatment. And so as we wrap up today, we'd love your feedback on today's session. So when the session ends, a brief survey will auto-populate on your screen, and that's your opportunity to provide your feedback about the session. And just know that your feedback is truly invaluable and helps us improve our future fireside chats and programming. So after you complete the survey, you can enter into a drawing to win a prize pack filled with goodies from the Masonic Cancer Center as a small token of appreciation for your time. And lastly, mark your calendars for our next fireside chat in recognition of Cervical Cancer Awareness Month on January 22nd from 1 to 2 p.m. So make sure that you follow the Masonic Cancer Center on all of our social media platforms for more details and to stay updated on future events and announcements from the Office of Community Outreach and Engagement. So thanks again, everyone, for joining. Happy holidays, and we look forward to seeing you in 2025.
Thanks so much. Take care.
Thank you. Take care.